Glycolysis is the metabolic pathway of breaking down glucose. So let's break down glycolysis. Keeping in mind that glycolysis, although it's an important pathway, it isn't the only fate for glucose. Depending on your cell type, you can hook up the glucose to form a storage form of glycogen. You could take it into the pentose phosphate pathway to regenerate your NADPH. If you need to combat oxidative stress or make lipids, make DNA and RNA and that sort of thing. You can take it in the hexosamine pathway if you want amino sugars, various options, but let's go through the process of glycolysis and let's see how it's very logical, no matter how scary it might seem at first. We have glycolysis, which is the breakdown of glucose to give you pyruvate. In the process, you're generating 2 ATP and 2 NADH. It happens in 9 steps. It starts with a 6-carbon compound, gets broken into 2 3-carbon compounds, and each of those 3-carbon compounds then goes through the rest of the pathway. So you have the bottom part of this pathway is taking place 2 times, and the top part is taking place 1 time. In the first step, hexokinase is going to trap the glucose inside of the cell by phosphorylating it. Phosphoglucose isomerase shifts things around so that you free up this end to be phosphorylated by phosphoglucose. You've now phosphorylated both ends. You can split it in half, and each half will be able to be interconverted, and each half will the phosphate group that's trapping it in the cell. That splitting is done by fructose bisphosphate aldolase, typically just referred to as aldolase, and everything after it in this pathway is going to be repeated twice. Once directly from this glyceraldehyde 3-phosphate that you made, and once from converting the dihydroxyacetone phosphate into the glyceraldehyde 3-phosphate using triose phosphate isomerase. And then everything down here is then going to get repeated with each of these 3-carbon pieces. The glyceraldehyde 3-phosphate is going to get oxidatively phosphorylated by glyceraldehyde 3-phosphate dehydrogenase. You stick a phosphate group on the other end. This is going to set you up so that you can transfer that phosphate group onto ADP to give you ATP using phosphoglycerate kinase. That's going to leave you now with 1 phosphate group again, but now you have a more oxidized compound, this 3-phosphoglycerate. Phosphoglycerate mutase is then going to shift this phosphate group to the middle position, which is then going to set you up for doing a dehydration reaction that kicks out of water and leaves you with a phosphoenol compound, specifically phosphoenol pyruvate. An enol is where you have a double bond between 2 carbons, and then one of the carbons in there is attached to an oxygen. This is highly unfavorable, and it much prefers to be in a ketone form where the double bond is from the carbon to the oxygen rather than between the 2 carbons. However, the phosphate group is trapping it in this state, and so we need to remove the phosphate group in order to get to the keto form. And when you do so, that's going to be highly energetically favorable, so favorable that you can stick that phosphate group onto ADP to give you ATP. And pyruvate kinase is the kinase that's going to help make this reaction happen, and it's going to leave you with pyruvate. The name's a little confusing because this reaction is very energetically favorable, so you're not just going to reverse it, but it's named in the kind of reverse as if you were putting the phosphate group onto pyruvate, but really what you're doing is taking the phosphate group off of phosphoenol pyruvate, sticking it onto ADP to give you ATP, and getting left with pyruvate. If we are doing aerobic glycolysis, that pyruvate is then going to get oxidatively decarboxylated by pyruvate dehydrogenase, give you acetyl-CoA, which can enter the tricarboxylic acid cycle. The NADH that you generated can transfer the electrons to the electron transport chain, pump out protons, and make ATP. In the process, you're regenerating the NAD+, and the NAD+ can then be used to keep glycolysis going. In the absence of the electron transport chain, in the absence of oxygen, you need another way to regenerate the NAD+ and a way to keep the NADH from getting high and inhibiting your pyruvate kinase and your other catabolic enzymes. One way that you can do this is by anaerobic respiration, anaerobic glycolysis. You take the pyruvate and you take your NADH and you stick the electrons from NADH onto pyruvate to give you lactate. That NAD+ is regenerated so you can keep glycolysis going. You can keep making the ATP from glycolysis, even in the absence of oxygen. However, if you're not doing the electron transport chain, you're going to be missing out on a lot of ATP. To go the kind of reverse direction and make glucose, you're going to actually have to use some different enzymes for the metabolically irreversible steps, this hexokinase step, this phosphofructose kinase step, and the pyruvate kinase step. The pyruvate kinase step, in fact, is going to need two enzymes in order to reverse it in gluconeogenesis and keep the gluconeogenesis pathway energetically favorable. At the same time, the glycolysis pathway is energetically favorable. The hexokinase step is going to trap this glucose in the cell. It has different fates. Phosphofructokinase is going to commit the cell to glycolysis. There are fewer fates after this phosphofructokinase step, so this is going to be very, very highly regulated. So there you go, the basics of glycolysis.